Now I'd like to introduce to you Sally. Sally is a PhD student who's working down at University of Otago, and her main interests are varroa sensitive hygiene, which I'm very familiar with, which is exciting, as well as the formworm virus and the effects that the virus has had on olfactory processing in the honeybee. So she is also the Apiculture New Zealand Conference Travel Award recipient for 2016. Thank you, that's very exciting for her. And she's also here with Jess Taylor and Rosanna Spears, who also got travel awards. So we'd like to welcome her to the stage. Thank you.
Thank you for the introduction, Michelle. As she said, I'm a PhD candidate in my third and hopefully last year of study as a student. I study at the University of Otago, and I am based at the Department of Zoology and also at the Department of Microbiology in the Otago Centre of Electron Microscopy. At microscopy and the electron microscopy unit, I study deformed wing virus. So I've been purifying the virus for single particle reconstruction, and also have been using the sample to determine the stability of the virus in various temperatures and pHs. At the Department of Zoology, I'm mainly focused on a behaviour called varroa sensitive hygiene behaviour. The talk that I'm going to be giving today is not about the adults that perform this behaviour, but about the brood cells that are targeted by the adults performing the behaviour. So I'm going to explain or remind you of one of the main mechanisms that honeybees use to resist varroa. This is a behavioural, hygienic behaviour that is targeted towards varroa. It consists of several steps. So first, the varroa is detected by the VSH bees under a waxed cap cell. Second is after these waxed cap cells have been detected, the cap is removed, which can be seen in the first photo. Lastly, these pupae are removed from the cell, as seen in the middle photo, and sometimes they are removed from the hive itself. Uncapping of these cells that contain pupa and varroa kills off the offspring of the varroa in these cells. It also forces the female varroa to exit the cell and infest another cell. Because a varroa the average reproductive cycle is about 4 in a female varroa, interrupting just one of these cycles can have a substantial effect. There's been lots of research done in this area, but the cues that are used by VSH bees to detect varroa under a wax cap brood remain unclear. There is a lot of evidence that points towards chemical cues as being an elicitor of this behaviour, but where these cues come from and what these cues are currently unknown. So we designed a behavioural assay. This allowed us to identify cells that are being targeted by VSH bees and cells that are infested with varroa but remain capped during the experimental time. So I'm going to take you through this assay. There are two hive types. The first is a varroa donor colony. This colony has an infestation rate of 10 to 35%. Our second hives are named VSH hives, and these hives have been bred for VSH behaviour. For the experiment, we take a capped brood frame from a varroa from the varroa donor colony, shake and brush all the bees off this frame, and leave it in the VSH colony for 1 hour. After this incubation period, we take this frame back into the lab and we identify the cells that have been uncapped during this time. These cells are of course infested with varroa. If they are not, then they are not collected as varroa targeted cells. When the targeted cells have been collected, we put the frame into the VSH colony, and this process is repeated over 2 days. At the end of 2 days, we collect our non-targeted cells. So these are cells on the same frame that are infested with Varroa but not targeted or uncapped during the experimental time. They are taken on the same side and in the proximity of the targeted cell that was collected. When the non-targeted cells have been collected, we get a new frame from the Varroa donor colony, and this whole process was repeated over several weeks. From these experiments, we found that targeted cells are indeed different to the cells that are not targeted by VSH bees. We found that the pupae that were targeted were developmentally delayed compared to the surroundings. So the non-targeted cells were within 1 pupal stage of its surrounding cells, whereas the targeted cells were 3 to 5 stages developmentally delayed, which means that these bees are likely to become dysfunctional adults if they get to emerge. Another finding that we found is that the brood ester pheromones coming from the targeted cells were different. The profile released from these bees were different, and they released 130% more brood ester pheromone compared to the cells that were not targeted by VSH bees. We also looked into the viral contents of the pupa in the samples. We found that deforming virus titers were not different between these 2 sample types. These 2 sample types were both infested with Varroa and had high titers of deformed wing virus, but there were no differences between the targeted and non-targeted cells. This was not the case for Kashmir bee virus, where we found that VSH bees selectively targeted high titers of Kashmir bee virus. The cues coming from the virus are not associated with the brood ester pheromone profile that I talked about in the previous slide. This shows that the results are at least part of the VSH behaviour is disease-specific, and that it involves multiple cues. So you'll see that I have only been talking about the pupae at this stage. But what about the Varroa content in these cells? The same behavioural assay was performed again. This time we were looking at the Varroa content in these cells. So we had targeted cells and non-targeted cells, and I also sampled 2 layers of surrounding cells. In these cells, I counted the number of females, the number of eggs, and of 2 instar stages of protonymphs and deutonymphs. I'm just going to mention now that in the surrounding cells, the grey inner ring and the darker grey outer ring will be separated, and this will become important in the next few slides. So there are 2 graphs, simplified graphs, that show the Varroa content in the targeted and non-targeted cells. In the first graph, you can see that the average number of Varroa offspring in the targeted cells is higher compared to the average number of offspring in the non-targeted cells. These trends were seen collectively, which is shown in the graph, and also in each individual category of Varroa. In the second graph, we can see that the average number of female Varroa is higher in the targeted cells than compared to the non-targeted cells. This indicates that there is more multiple infestation in the targeted cells. In a cell, there can be 1 Varroa, and I've seen up to 17 Varroa in a single cell. And the graph shows that in targeted cells, there are almost twice as many Varroa in a cell compared to the non-targeted cells. If we now look at the surrounding cells of the targeted and non-targeted cells, we can see that the proportion of cells with Varroa surrounding the targeted cells is much higher compared to the proportion of cells with Varroa surrounding the non-targeted cells. Also, in the second graph, we can see that the average number of female varroa is again higher in the targeted cells compared to the non-targeted cells. If we now separate out the targeted cells into the inner ring and outer ring, you can see that the inner ring of targeted cells had a higher proportion of varroa compared to the outer ring of targeted cells. In the second graph, you can see that the average number of female varroa in the inner ring of targeted cells is higher compared to the outer ring of targeted cells. Both of these trends are not seen in the non-targeted cells. From the graphs that I have shown you in the previous 3 slides, we can see that the cues eliciting VSH are associated mainly with the female varroa, but also there is input from her offspring. We think that this may be caused by multiple feeding sites. So when a varroa enters a cell and the cell is capped, she establishes a feeding site that she feeds off, and later her offspring will feed off as well. So if there are multiple females in a cell, this means that there are more feeding sites on this pupae that could induce it to release stronger cues that elicit VSH behaviour. Another reason is that multiple females produce multiple offspring. So the stress responses in these cells with multiple females could be stronger than that of singly infested cells with only one female varroa. We could also see that highly infested patches are more likely to be targeted by VSHBs, and that there were more varroa in the inner ring of targeted cells compared to any other ring. This indicates that the immediate surroundings have a greater effect on which cells are targeted by VSHBs. It has been reported in literature that VSHBs sporadically uncap uninfested cells. From these results, it is possible that VSHBs detect a highly infested patch of brood and randomly select a cell within this patch to perform on. And this could explain why VSHBs sporadically uncap uninfested cells. So over the course of my PhD, part of my research has been investigating how VSHBs determine which cells to select and sacrifice. This question is central to the success and survival of varroa-infested colonies. The research presented characterises brood cells from the content within the cells and also with the surrounding cells. We believe that the results seem promising, and with further research, we hope to aid detection of VSH hives and also selection of bees based on the VSH trait. I would like to acknowledge Alison Mercer, who was my supervisor at the Department of Zoology, Binia Bostina and Vernon Ward, who are also my supervisors, Fanny Monday, who I collaborate on with a lot of my research, and France Last from Beta Bees for providing the hives that were bred for VSH behaviour. I would also like to acknowledge the Travel Award by APINZ for giving me this opportunity to talk to you and helping me get here, and also funding from the University of Otago. Thank you. I think we have time for one question.
Thank you. I think it's a really good work. And
Thank you.
have you thought about to stimulate the VSH behavior, perhaps some of those cues from the...
Sorry, could you repeat the last part?
You say that some of those cues could be those esters, pheromones.
Mm-hmm.
Have you thought about there may be ways to stimulate the VSH behavior by using some of those pheromones?
Yes, we have, along with this research, we have identified odours that are coming from these cells. And this research was conducted 2 years ago, and earlier this year we performed an assay by collecting VSHBs and testing these odours on them, and also collecting non-VSHBs that are at the same age of the VSHBs that we collected and testing the same odours and to see how they perform. These experiments were run over a long period of time, and we're still in the process of looking at the videos and analysing them. So we have been taking these cues and odours and testing these out as well. Thank you.